Hello, this video is going to go over chapter 23, the digestive system, objective 3. So with this objective, we're going to discuss the processes and control of ingestion, propulsion, mechanical digestion, chemical digestion, absorption, and defecation. So when we're talking about nutrient digestion, I've got several videos hyperlinked within this PowerPoint to kind of help you out with that. This one's about 3 minutes long. There's a little bit of inconsistency at the very, very end of this where it talks about the lipids, but other than that, it's a really good visual on how nutrients get digested. So just a reminder, we've mentioned this previously in a prior objective. So with the digestion of all nutrients, this right here is the lumen of the small intestine. You've got proteins, carbohydrates, and fat. They're going to pass through this epithelial cell. Remember, this is going to be a single layer of column-shaped epithelial cells into either the capillaries or the lymphatic system. So here is a list of digestive enzymes, where they go, where they're coming from, what their job is. I suggest you really take a look at this particular one and study this. So let's just kind of talk about it real quick. So for example, salivary glands. Salivary glands are located in the mouth. Their secretion is saliva. Now within that, there's this enzyme called salivary amylase. And again, I'm going to kind of help you out with this word here. Amylase is the name of the enzyme. You know it's an enzyme because it ends in ase. Where does the amylase come from? It comes from the salivary gland. So you've got several different kinds of amylase throughout this list. Pancreatic amylase is another one. What's going on with pancreatic amylase versus salivary amylase? It's still amylase, it's just telling you where it's coming from. So that's all that is. So that's the distinction. What is the purpose of salivary amylase? What does it do? It breaks down carbohydrates. Saliva helps ease swallowing, it gets everything moist, but the amylase part of it is the breakdown of the carbohydrates. Now, your gastric glands. Gastric glands are going to be located in the stomach. We've already talked about a few of these. They secrete some gastric juice. What is the enzyme involved? Well, you've got hydrochloric acid, which comes from the parietal cells. You've got pepsinogen, if you remember, was inactive, coming from the chief cells. Now, when pepsinogen and hydrochloric acid are in the same place, the hydrochloric acid activates the pepsinogen and turns it into pepsin. So pepsinogen by itself, nothing. Hydrochloric acid is not going to be there without the pepsinogen, but it's going to help break down proteins. But the pepsin, the combination of the two, create this chemical called pepsin. Now, what does pepsin do? It breaks down proteins. So hydrochloric acid denatures proteins. We talked about this in a previous chapter, I believe it was chapter 3, where we talked about characteristics of proteins. And if you put them in a very acidic environment, low pH kind of situation, it's going to break the protein down and they're not going to be able to work ever again. So it's going to break down the protein, and then the enzymes, the pepsin, is going to break the protein down into its building blocks, those amino acids. Also, you're going to have mucus that's being secreted from the goblet cells, and that's going to help protect the stomach, to protect the stomach lining, so you don't get ulcers and things like that. Now, the pancreas has got a lot going on. The target organ, it's going to be putting those secretions into the small intestine. It's collectively known as pancreatic juice. So what is involved in this pancreatic juice? Well, you're going to have pancreatic amylase. So you're going to have amylase be secreted again. Pancreatic lipase, that's going to be an enzyme that's going to break down lipids. And then bicarbonate. Pancreatic lipase is going to be part of chymotrypsin, peptidase, and trypsinogen. So those are the three that make that up. Now, what is the bicarbonate for? The bicarbonate neutralizes the acid. So let's take a look and see where this just came from. We just dumped everything out of the stomach into the small intestine. We just dumped everything out of the stomach into the small intestine. Now, what was in the stomach? Hydrochloric acid. So now you're putting the hydrochloric acid into the first section of the small intestine. That's not good. So to get it back to a normal pH that's not as destructive as that pH of the hydrochloric acid in the stomach, bicarbonate is secreted by the pancreas to kind of neutralize it, bring it back up to a pH of around 7. Now, you also have all these pancreatic enzymes, the amylase and the lipase and the peptidase. These are all returning and jumping back into the small intestine to break down carbohydrates, fat, and proteins. And you may be thinking to yourself, okay, that's great. What happened to that salivary amylase? I thought we already kind of digested that. You did. You started the process of carbohydrate digestion, but as soon as that amylase hit the stomach, that darn hydrochloric acid broke down all of those proteins. Enzymes are proteins. So the amylase that was there isn't working anymore. So you have to add it back again. So now that the pH is where it should be in the small intestine, that pancreatic amylase will now work to continue the breakdown of those carbohydrates. The liver and the gallbladder together are going to be secreting bile. Now, the liver has a lot of jobs to do. So what it does is it makes bile and sends it to the gallbladder for storage. Okay, it also secretes a little bile in and of itself. But it does send it to the gallbladder for storage. So when the food comes through, the gallbladder will secrete that bile into the small intestine. That small intestine bile is going to emulsify fat. A fancy word for it's going to, remember, fat doesn't like water. So when the fat is in the small intestine, it likes to find other fat molecules to hang on to, so it forms this big floating glob of fat. Kind of like if you were to take oil and water and shake it up, you would see all these little bitty oil particles, but then if you let it sit long enough, it will reform that big, huge, original, all-together blob of oil that's going to be floating at the top. So what does bile do? Bile keeps the fat from coalescing and coming back together again. It keeps it in small little globules so it can't come back together and form that big blob. So that's what emulsify means. And then your intestinal glands, you're going to have glands within the intestine. Small intestine specifically, you're going to get these intestinal juices. This is one of the things is it's going to convert chymotrypsinogen. It's going to secrete peptidase and trypsinogen to chymotrypsin. Some peptidase, which is going to break down proteins, and trypsin. Some amylase, so you're going to have some lipase as well. So you've got your same molecules that break down all of your macromolecules that are going to be yet secreted again. So yes, your pancreas secreted them. Now your small intestine is also going to be secreting those same enzymes. And it's for the intestinal breakdown of carbohydrates, fats, and proteins. So each time you're just getting the macromolecule smaller and smaller and smaller, and you want it to be actually in those building blocks. You want them to be that small by the time they get to the intestines so they can pass through that wall. So you just keep secreting these same enzymes to keep breaking that food down. So here's a handout that's in your packet. I really like this one. It's maybe hard to see because it's a black and white picture, but here you can really see the colors and it shows the white dots. So what's going on here is it shows you, for instance, this is the mouth. And take a look at what's happening here. You got a lot of white dots in the carbohydrate zone. What that's telling you is you got carbohydrate digestion that is beginning in the mouth. Carbohydrate digestion begins as those salivary glands are secreting amylase, and that's going to start in the mouth. Now, notice we have a little bit of a blip over here with the fat. You're not actually digesting the fat. What you're doing is it's actually melting. So it's kind of more mechanical, I guess, in a way. But if you were to take like a piece of chocolate and put it in your mouth, it's going to melt the chocolate. It's not really going to break it down. It's just going to melt it. So it's turning it from a solid to a liquid. Now, as you move into the stomach, look what happens. We've got carbohydrate digestion is going to continue. This is when the chyme is still in the upper portion of the stomach, that fundus, before the hydrochloric acid has set in. Notice you have this area right here where nothing is happening. That's because the amylase that came from the mouth, those salivary glands, it's been destroyed, so we have no more carbohydrate digestion. Fiber, still nothing digested at all. Protein, look, this is where protein digestion begins, in the stomach. Why? Because of that hydrochloric acid that is present there, along with the pepsinogen. Notice there's also no fat absorption or no fat breakdown that happens in the stomach. Now you get to the small intestine. So now we've got the enzymes that are coming from the pancreas and from the small intestine themselves. Look, you have got all kinds of digestion happening in the small intestine. All those enzymes have returned, and we have got the breakdown of all of our 3 macromolecules continuing in the small intestine. Look what's not being broken down: fiber. Remember, fiber is made up of cellulose, and when we talked about back in chapter 2, cellulose was a type of carbohydrate that we cannot break down. So fiber doesn't get broken down at all, but is necessary for the health of the small intestine and the digestive system. The large intestine. So you're going to have some fiber that's going to be able to get absorbed, but not most of them are going to pass right through the large intestine and go straight into the excretion part. So again, the large intestine's main job is fluids, and some minerals are absorbed. So again, this is the main location for water absorption, remember? So again, a good little visual of what's going on, and it does break down all of the macromolecules and what's going on with them as they move through the digestive system. And this is just a close-up picture of each one of those images, so that way you can see it a little bit better if you didn't have your handout with you. Most of the absorption of molecules from carbohydrates, proteins, and fats takes place in the small intestine. We just saw that. The small intestine is 10 foot long, but it has the surface area of a tennis court. So what is going on with that? If you were to take a look at the inside, this is the lumen of the small intestine. If you were to take a look at that, those walls are not smooth. You have got these large circular folds, and within those circular folds, you have these villi. All of this is increasing the surface area. So the purpose of that is to get the greatest absorption of food, and I shouldn't call it food at this point, through that lining of that small intestine. So you can see here that inner surface of that small intestine. It appears smooth, but it has hundreds of folds which increase the surface area. Each fold has these thousands of finger-like projections called villi. And then even the villi are composed of hundreds of cells, each covered with microvilli. All of this helps increase surface area. The crevices between the villi secrete those intestinal juices. We've already looked at that, and those goblet cells secrete mucus. Each cell of the villi, again, like we said, is coated with thousands of microvilli. An
example of how enzymes function.
There's a video here for you to watch. It's about 2 minutes long. You're welcome to take a look at it just to kind of get a better visual. And it actually goes through the breakdown of sucrose. So it kind of uses that as an example. Within the microvilli, there are hundreds of different enzymes which recognize and act on different nutrients. So a lot of stuff going on there. When a nutrient molecule has crossed the cell of a villus, it enters either the bloodstream. Now this is going to be for carbohydrate breakdown and proteins. So the building blocks of carbohydrates, which would be sugar, glucose, and proteins, which would be those amino acids. And then the fats or the lipids will enter into the lymphatic system, those triglycerides. Both transport systems supply vessels to each villus. So in other words, around each villus, let me see if I can go back a minute. Take a look right here. You have got your capillary, artery, and vein, and then you also have your lymphatic system. That lymphatic system is going to be represented in green. So molecules that enter the system and where they're entering at. So in the bloodstream, you're going to get your water-soluble nutrients, such as the byproducts of those proteins, the amino acids, and the byproducts of those carbohydrates, sugars. That's going to go into the bloodstream. Those substances from the bloodstream are going directly to the liver. So the next stop, once those substances have entered the bloodstream, liver is the next stop. Now, the lymphatic system, your fat-soluble vitamins and your fats are going to enter into the lymphatic system. Now, these are going to, as they pass through the lymphatic system, they're actually going to enter into the blood supply around the heart. So they're going to stay in the blood supply until they get to the heart, and that's when they're kind of jump back into that blood supply again. So they will get into the blood supply eventually, but they're going to travel through the lymphatic system until they get to around the heart, and that's where they're going to jump into one of those veins. So you've got this handout in your packet there. It really kind of shows the blood flow, including this little section right here on the liver. So this is what I really wanted to show you here, this focus right here on the digestive tract and the liver. So you've got your right side of the heart, you've got your oxygenated blood, capillaries are going to drop off oxygen, pick up carbon dioxide. Veins, left side of the heart is going to be deoxygenated. Now, if you take a look at number 1, blood leaves the right side of the heart by way of the pulmonary artery. So we're going to go up the pulmonary artery, go into the lungs, pick up that oxygen supply, go back to the left side of the heart, go back to the left side of the heart, and then get pumped out. So here we're at number 3. The blood leaves the left side of the heart by way of the aorta, and then that's going to end up going to the body. So if you follow that along, we're not going to go up, we're going to go down. So we could either go up to the head or we could go down to the lower body. We're going down to the lower body for this discussion. If you take a look at number 5, blood can go to the digestive tract and then to the liver, or it can go to the pelvis, kidneys, and legs. So we're going to take the little path that's going to take us to the digestive system. Bottom line here, so that blood is going to go to the digestive system, and at this point, it's all wrapped around the small intestine, and it is going to now be picking up all of those nutrients, okay? So it is going to then pass from the digestive system and head over to the liver by way of what's called the hepatic portal vein. The hepatic portal vein is going to carry nutrient-rich blood, but notice it doesn't have a lot of oxygen in it. It's kind of turned purple here. So it's a capillary, but it's kind of like it's dropped off its oxygen supply, but it hasn't quite, it really didn't do a whole lot of gas exchange. It did a little bit, but not a whole lot. So that's why it's kind of purple. It's kind of mixed, half and half. But that hepatic portal vein is going to take that nutrient-rich blood to the liver. And then the liver is where you're going to get some more gas exchange going on. Then it will completely be deoxygenated, go through the hepatic vein, back into the inferior vena cava, and into the right side of the heart. So again, this is that close-up here that I wanted you guys to see. And it's this path right here. That oxygenated blood is going to go to the digestive system. From there, you're going to get the nutrients, specifically the breakdown of the carbohydrates. You're going to get your breakdown of your carbohydrates and your breakdown of your proteins. Amino acids are going to enter into the hepatic portal vein and go to the liver. So it's going to the liver. Now, we also did not mention in the previous slide, your lipids are going to go through the digestive system and go into the lymphatic system and they're going to travel back. Let me back up a slide. They're going to travel back up, and notice we said they were going to enter in around the heart. They were going to jump back in and jump into that blood vessel around the heart. So you can kind of see number 7 there. Here's another picture showing you kind of the same thing here. Vessels are going to gather nutrients from the digestive tract. So this vein right here, all these are blood vessels, these blue right here. They're picking up the nutrients from the digestive system, which you're not really seeing a whole lot of here. A lot of that small intestine has been removed, but it's entering into that blood vessel. into the capillaries, and then all of those nutrients, that nutrient-rich blood, is going to travel by way of the hepatic portal vein to the liver. Now, keep in mind what's the nutrients that we're talking about here. It is carrying the breakdown of carbohydrates and the breakdown of proteins. That's what's in that hepatic portal vein. It is going to the liver, okay? The vessels merge into the hepatic portal vein, which conducts all absorbed materials to the liver. The hepatic artery is bringing oxygenated blood, so that is this blood vessel right here that is bringing oxygenated blood to the liver. We talked about that in the blood vessel unit, but it doesn't have any nutrients in it. It's just rich in oxygen. A large network of capillaries branch all over the liver, making nutrients and oxygen available to all of the cells of the liver. The hepatic vein gathers up blood and returns it to the heart. So this hepatic vein right here is taking all of now the nutrient deficient and oxygen deficient blood back to the heart. So that's what's going on in that hepatic vein. Keep in mind, you've got a lymphatic system that's carrying the lipids, and that's not going here. Now, carbohydrates. Pretty good little video on carbohydrate digestion. I encourage y'all to take a look at that. Another video here on carbohydrate digestion in case you wanted to see another one. This again, carbohydrate digestion. So I'll put some videos up to kind of help you visualize what's going on there. Protein digestion. You've got your proteins that are coming in. You're going to eat them. They're going to go into the stomach where they will get broken down with the presence of hydrochloric acid and pepsin. They will then go to the small intestine where they're going to continue to get broken down by peptidases, and then they will be actively transported from the lumen of the small intestine into the bloodstream. Again, a pretty good video on protein digestion. Now for fats, we mentioned previously that fats melt in the mouth, so they just go from kind of a solid more to a liquid phase. Infants actually produce this enzyme called lingual lipase to help digest those fatty acids and things like that that are found in milk. So infants have that ability to make that protein. Adults do not. In the stomach, gastric lipase is going to be secreted. So some digestion of fat occurs, but not much. The main amount of fat digestion is going to occur in the small intestine. So that's the main place lipids are going to end up breaking down. So these fats in the stomach become smaller fat droplets. Most, again, of the digestion of fat occurs in the small intestine. The large intestine, fat and cholesterol that is trapped in fiber is going to exit in the form of feces. So the goal of fat digestion is to dismantle triglycerides into small molecules that the body can absorb. Those small molecules are monoglycerides and fatty acids and glycerol. So that is what the goal of the breakdown of fat is, into monoglycerides, fatty acids, and glycerol. Now, in the small intestine, fat enters the small intestine and triggers the release of cholecytokinin. That's abbreviated CCK. It's a hormone that kind of lets the gallbladder know when to release bile. Again, nothing happens without somebody telling it what to do. I've said that a bazillion times. So how does the pancreas know it's supposed to release, or how does the gallbladder know it's supposed to release bile? Well, cholecytokinin shows up. Bile is an amphipathic molecule. That means it's got a hydrophobic part and a hydrophilic part. We talked about this when we mentioned phospholipids. Remember, they were also amphipathic. So bile acts as an emulsifier because it has an area that likes water and an area that hates water. Pancreas secretes lipase, which actually digests the fats, breaks them down into smaller pieces. Most of the breakdown of fat, hydrolysis is the breakdown of fat, occurs in the small intestine. The major fat digesting enzyme is pancreatic lipase. And I just put down how to pronounce cholecytokinin down here. That's all that was. So we talked about how you've got your triglyceride. So you've got your glycerol molecule right here. That's your glycerol molecule. And you have your 3 fatty acids. That's why it's a triglyceride. Fatty acid, fatty acid, fatty acid. And what digestion does is it breaks it down into a monoglyceride. So this right here is your monoglyceride. And then you have your fatty acid. And then you have another fatty acid right there. So that is your monoglyceride and 2 fatty acids. So what happens to the bile? Bile enters into the small intestine from the pancreas. Once it emulsifies the fat, it can go 2 different places. I tried to show y'all what a vial of bile looks like. Bile is green. And if you notice, that's why the gallbladder underneath the liver, when you see it on models, it's green because bile is green. Most of the bile, you need to know the 2 destinations, most of the bile is resorbed from the small intestine and recycled. So you get to use the bile over and over again. But some of it, like we said previously, just a few slides back, can get trapped in fibers in the large intestine and get excreted in feces. Now, lipid absorption. Some molecules, such as glycerol and short to medium chain fatty acids, can go easily through those intestinal cells and go directly into the bloodstream. Larger molecules, such as monoglycerides and long chain fatty acids, are emulsified by bile, forming spherical complexes called micelles. So here is that fatty acid molecule right there, that long chain fatty acid molecule. And if you take a look, it is organized into this structure called a micelle. So it's just a circular structure of a bunch of fatty acids. Micelles diffuse into the intestinal cells and get reassembled into new triglycerides. Seems kind of silly. We're taking a triglyceride, we're breaking it down, and then we're building it back up again. So it seems kind of silly. Triglycerides are too big. They can't pass through the small intestine wall. So you got to break them down into small pieces, and then you reassemble them afterwards. So these new triglycerides get packed into vesicles called chylomicrons, which get into the lymph. And then they enter into the bloodstream, specifically at the thoracic duct. The thoracic duct is part of your lymphatic system. That thoracic duct is going to dump its contents into the subclavian vein. So right there is where it goes from the lymphatic system to the circulatory system. If you remember where your subclavian vein is, it will then go straight into the heart. So this right here is what those triglycerides look like. So you've got this chylomicron here and all these triglycerides inside. The blood is going to then, once it gets into the subclavian vein, the blood is going to carry the lipid to the rest of the body for storage. So again, just to kind of see that picture a little bit, you've got your thoracic duct right here, that green one, which is taking it, dumping it into the subclavian vein right there. That subclavian vein will be entering will be going into the heart by way of the superior vena cava into the right atrium. So just to see a picture here, you've got your fatty acids and monoglycerides. They're going to be emulsified by bile, so notice they're getting smaller, right? That bile has come along, represented by those little sticks right there, and it's going to keep that fat from reassembling into these big, large globs. And then they're going to form micelles. So you have these little bitty particles right here. Those micelles are going to be able to pass through the small intestine lining. The fatty acids enter the epithelial cells and reassemble into triglycerides. Those triglycerides combine with proteins inside the Golgi body and form a chylomicron. So we have this chylomicron right here. Those chylomicrons are going to enter into the lymphatic system, pass through that lymphatic capillary right there, and off to the thoracic duct it's going. And that concludes Objective 3. I know that was a long one, so sorry about that. We'll do Objective 4 next.